It is Tuesday, June 16th. What? Jeff, you're throwing off my vibe. It is June 16th, Tuesday, and you are watching the Biotech Voyager. It's the show that covers what's next in biotech, the signals that you're not really hearing about anywhere else that deserve more attention just because of how big they could be in the future. Jeff, we got an exciting show today.
Yeah. Sorry to throw off your vibe. I love the confused response. You're just like, what? wait, what's happening with Jeff? I couldn't help myself. The futuristic music just turned me into an alien, I guess. I don't know.
Yeah. Brand new music intro. More high quality stuff to come, folks. All right. So, Jeff, a lot of different things to talk about today, but yeah, let's obviously start off with the early stage biotech leaderboard that you can all go and access at thebiotechvoyager.com. What are some of these early stage companies that we should be looking at? Right up at the top here is Aerialis Therapeutics. We'll get to them a little bit later, but maybe we can just touch on them quickly. What's really pushed them to the top here?
Yeah, so they shot up, I think last night they shot up, maybe 2 days ago. But they just pushed into phase 2. They got an IND cleared for phase 2 for one of their assets, and they dosed their first patient in a phase 2A for a different asset. So we'll get to what they're doing, but their big jump was 2 really big phase 2 enabling events.
Excellent.
Yeah, pretty cool stuff with using antibodies to quench autoantibodies for neuropsychiatric disorders. Pretty interesting. But then of course, Alpha Tau is still here. They've just been super active lately. And Viva still here with their CAR T cell stuff. We'll get to these companies, but a couple of the ones that we're going to touch on that I'm really excited is companies like ANACA TCR-T cell company for solid tumors dosed their first patient last week. Really good stuff. I think we're going to talk about Apligon. You've got Apligon. And things I think we had one more on the top 15 here. Let's go to the recent signals, maybe. Definitely, yeah. So let's just jump into it. What's the first one? Enterome? Let's pop up Enterome here.
Yes.
So Enterome, I think it was last week, maybe Friday, or it was yesterday. So Enterome presented phase 1 data, I think, phase 1/2 data for their oncomimetics. And it gives me a reason to talk about oncomimetics again. We've mentioned Enterome in the past before, and we'll go through the data, but I really, really like this approach. And I dug in a little bit more to what makes this approach really interesting. Or I dug in to figure out what it was, and I found out that there's something about it that I wasn't really considering that makes it really interesting. Okay.
Well, first of all, let's back up for a second. What in the world was that massive word that you just used that only 4 people are going to recognize?
Oncomimetics?
Yeah. What the heck is that?
Yeah.
You got to back up for a sec, Jeff. That's 5 syllables.
Okay, so perfect. So basically what Enterome is doing is they're a cancer therapy company. And they make, technically speaking, they make cancer vaccines, peptide-based cancer vaccines. Cool. Right? But they use peptides that are derived from bacteria, from microbes, and those peptides mimic oncogenic peptides. oncomimics, oncomimetics.
Okay, here we go. We're
Yeah.
breaking down the word structure. I like it.
Yes.
Okay.
So
Yep.
they're peptides derived from bacteria that will basically trick our body into killing something that is present on cancer cells.
Interesting.
Yeah.
So
And what makes
the
it
question there, is there like a difference in like the epigenetic profile since this is made in bacteria versus like a similar peptide that's being expressed in cancer cells, and that's kind of what forces the immune response, or are they like functionally identical?
they're basically functionally identical. And the word that is really important here is cross-reactivity.
Okay.
Cross-reactivity, the word is kind of self-explanatory. Cross-reactive. Reactive in an immune sense, meaning the T cells can react to it and our body can react to it. And then cross, meaning we have a response for peptide A, but that response for peptide A can also attack peptide B or peptide C. And there's that cross reactivity. I use the analogy of, I think last time I used my phone and this remote control, right? Like if I have my phone here and our body wants to kill my phone, right? You can imagine a situation where like, let's just pretend that this has some kind of similar characteristics to my phone. And because my body's trying to kill my phone, it will serendipitously also kill my remote control. So now you can picture that this is the microbial derived peptide, and here is the peptide on the cancer cells, and they'll kill them one and the same.
Yep. Cool.
Now, what's interesting, I hear you asking, what's so interesting about this, Jeff? What's so interesting about this, Jeff, is that the oncomimetic peptide that Enterome just got data for is being used for B-cell malignancies. And this oncomimetic peptide actually mimics CD19, CD20, CD22, and the BAFF receptor.
Yeah, those are some pretty big cancer targets. Let's go.
Yes. And the thing is that if you wanted to have like a CAR T cell target all four of those things, you would need to make a CAR T cell that it's like a tetra-specific CAR T cell, right?
Yeah.
But with this oncomimetic, because it mimics the epitopes that are present on all four of those, one single oncomimetic or one single cocktail of this oncomimetic will actually create reactivity against CD19 positive B cells, CD20 positive B cells, CD22 positive B cells, and BAFF positive B cells, which means good luck doing antigen escape cancerous B cells. So with one single like either cocktail or one single oncomimetic protein, they can get this tetra-specific immune response.
Super interesting. Okay. And again, right, with something like if I were to go with a CAR T approach for this, I would have to come up with some sort of CAR receptor that's able to bind all four of those different things. But this is just being like, hey, let's use the existing equipment we've already got inside of the biology, and let's just teach them what these four different key cancer targets are.
Exactly. And the thing about Enterome that I really like is that if you were to try and give let me give you a pop quiz. You ready?
Go.
If I gave your body a CD19 cancer vaccine, what would your body do?
It would ideally generate antibodies against CD19.
No, it would not. Because you have tolerance for CD19. Your body should not be reacting to CD19 naturally. It should not do that. If it did, you'd be dead.
Fair enough.
That would be a lethal autoimmune disease, right? What Enterome is able to do is they can divert tolerance because these are bacterial-derived peptides, right? So we don't have any tolerance for these things. But then that cross-reactivity leads to our body can create an immune response because there's no tolerance there.
Yeah.
But now that we had that immune response, it's going to be able to target CD19, CD20, CD22, and BAFF.
Is this a problem later on once I'm cancer-free?
No, not any more than a CD19 targeting CAR T cell would be.
Sweet.
You get B cell depletion, and then after a while, this vaccine will just kind of dissipate, right? The immune response will go down, and I guess If there may be a problem there. Yeah, because
we might be missing something.
No, they're definitely, yeah, well, there would need to be some kind of like recapitulation of the B cell population eventually. So I'm wondering if Enterome speaks to that. Because here's the problem. You've pointed out the problem, right? With a CD19 targeting CAR T cell approach, for example, or rituximab, a CD20 targeting antibody, you can administer the T cells or you can administer rituximab and you will get a temporal depletion of the B cells. Eventually, the CAR T cell population dissipates, or eventually the rituximab will go away and you can have like clearance for the B cell population to emerge again, right?
Right.
But if you give your body a vaccine, that immune response will generate memory, and then you will never be able to have repopulation of your B cells. Because the second that you do, that you're going to have that immune response again. So there must be something that they can do to stop their immune response.
Yeah.
And like get rid of that memory. I don't know.
Who knows? But yeah, regardless, right? They are going into what, phase 2? Is it? Or no,
they presented some data. So this won't. Oh, it will show perfectly.
Sweet.
So let's see if any of so positive immune responses were confirmed in 43 of 48 tested patients. And again, this is in B cell malignancies, maybe some kind of like B cell lymphoma. They got CD8 positive T cell responses, and they got these CD8 positive T cells, the B cell specific T cells, detectable up to 34 months. So that's over 2 years, almost 3 years, which is
sweet.
That's the problem though, right? Like that would be a problem. You wouldn't want to have a B cell depleting T cell population in your body for 3 years. That's actually insane.
There's got to be something we're missing here.
Yeah, I'd love to talk to the team to figure out
benefit to the patient.
Yeah, and it would be really good for cancer, but it would also be good. This approach would be good for autoimmune disorders as well, right? B cell depletion is used in autoimmune disorders as well. But I'm just wondering, how do they ensure a B cell repopulation?
I don't know, but it's interesting nonetheless. We'll see.
Yeah.
Cool. Well, folks, if you have been reading the banner along the bottom, we are going to have a guest who raised a boatload of money recently to go after non-viral gene therapy, Ken Greenberg from SonoThera. We've covered them on the show in the past. And in fact, one of our most popular clips was talking about the approach that they are taking, which is they're going to be using ultrasound to deliver full genes into the body using these really cool microbubbles. So he's going to be coming on at noon EST. So if you stick around for that, feel free to have some questions primed and ready for him when he comes on. But yeah, excited for that conversation. Excellent. Okay, so if we're moving on here from Enterome to Aerialis Therapeutics, I believe this one is mine, Jeff. But yeah, first of all, let's just kind of go back for a second. They jumped to the top of the Biotech Voyager. Can we go take a look at that?
They did. Indeed.
So yeah, they jumped right to the top of the Biotech Voyager, massive signal inside of the space. And so let's kind of break this down, why it is that they've generated so much buzz. Okay, so first of all, so again, kind of the way this graph works, you see the little points on it. It showcases the different signals there. So if you hover over there, yeah, so they have dosed their first patient in a phase 2A, and also they got FDA approval for a trial as well, right? Is that the one that's in Korea, or is this a separate trial?
Let's find out. So this is the FDA. It's one announcement, right? Okay. So they cleared. So it says first patient treated with ART-5803 in open label phase 2 study, and then FDA clearance of IND for randomized phase 2 study. The first patient dosed with their anti-NMDA was the one in Korea. And then they have the phase 2 approved for the US.
Okay, nice. Okay, so there are 2 separate trials for the same therapeutic going on.
It appears so, yeah.
Nice. Okay.
One in Korea, one in US.
Nice. Okay, so yeah, let's dive into their approach here a little bit. So they're going after a disease called anti-NMDA receptor encephalitis. That is a mouthful. So in plain terms, the patient's own immune cells make an antibody that attacks a key receptor inside of the brain. So this is an autoimmune play, a pretty nasty one. Autoimmune inside of the brain, never a good time.
Not good.
I imagine this is probably pretty fatal pretty quickly or it's going to be severely neurodegenerative. I don't know.
I think it has to do with like
but
neuropsychosis disorders. Not necessarily like lethal in the traditional sense, but things like schizophrenia, dementia, autoimmune related psychosis. So I'd even argue this is really rough stuff. Like is like, this might sound kind of inflammatory and controversial, but I would rather die. It's like, don't give me like really, really bad psychosis and schizophrenia. I feel like to a certain extent, the lethal neurodegenerative disorders, neither are good.
Yeah.
Let's just put it that way.
Regardless, yeah. So a really nasty disease. And so the approach here that they're working on is the mechanism. So most of the autoimmune drugs in this kind of area shut down the immune system broadly. However, what Aerialis Therapeutics has developed is an antibody that physically blocks the bad antibody from reaching the receptors. So it's a targeted block here. Instead of kind of going in and trying to do a bunch of sledgehammering, you can just basically have an antibody that goes in and just basically stops the bad antibody from being able to bind. And so therefore you eliminate the symptoms associated with this disease, ideally, and you don't get the whole autoimmune nastiness and the psychosis that comes along with that. What a nasty disease. Like gross.
Yeah. So this is actually very interesting because neurological autoimmune disorders, things like you can think of like multiple sclerosis is like one of the most traditional ones. A lot of the therapeutic approaches have involved two major things: systemic immunosuppression, blanket, right? Or the inhibition of immune cell trafficking into the brain, right?
Yeah.
Both are relatively non-specific to the disease.
Yeah.
Right? You're just like saying, okay, immune system completely shut down, or immune system get out of here, right? What I've seen more recently, and with some other like in some different emerging CNS therapeutics companies, is because of our increased understanding of how these diseases work and our increased ability to get powerful therapeutics across the blood-brain barrier, we are now seeing much more targeted and specific like neuroprotective therapeutics. So blocking a specific receptor that's engaged in the pathology, or, you know, and a lot of these things are caused by relatively normal neurotransmitters that just kind of go awry. So like in multiple sclerosis, you get a lot of like glutamate signaling, and glutamate is really important for neurotransmission. So just blocking pan-glutamate receptor blockade is not good. It's not really good. But being able to block a specific pathological associated receptor, that's much better. But finding the biology, getting the biology right, and then getting the actual specific therapy, like very target specific therapeutics into the CNS has been a challenge. But with advances in research and, you know, advanced therapeutics technologies, it's becoming more possible. And this is just one more iteration of that, where instead of just like blocking the immune system, they can actually have these antibodies. It's got antibodies here. And this is interesting too. It's a single-armed monoclonal antibody that competitively inhibits the NMDA receptor autoantibodies to treat anti-NMDA receptor encephalitis. This part I think is interesting here. Single-armed monoclonal antibody. I am wondering if this is because it makes it smaller.
Makes it smaller, but then also you kind of imagine that's going to be a bit more of a reasonable shape for acting as a blocker, essentially, right?
Maybe. It would depend on the actual like biochemistry of the receptor and or the ligand. But I'm thinking that it might actually be a method by which they cross the blood-brain barrier.
Okay.
If it's smaller, possibly, because it will still have the FC region and then one arm. So it's still pretty big, but like I know that like nanobodies, for example, are more able to cross the blood-brain barrier than an antibody is.
Yeah.
So I'm wondering if the just cleave off one of the arms and you still get specificity, but it's now like a third of the size.
Yeah.
Or it's
That'll
2/3 of the original size.
And again, right, it's designed not to be functioning as a normal antibody. Its goal very much is just basically to be something that sticks in the way.
Yeah.
And you've kind of got the minimal amount that you need to kind of sculpt it into that little pocket that the bad antibody is trying to get to. And you're off to the races. But yeah, really exciting to see. Like, again, for something like this, it's a rare disease, no existing treatments whatsoever. Horribly nasty psychosis. Really exciting to see something like this moving along through the clinic with some pretty great signals. Yeah, like what else are they building? So I imagine that's the first one. So they're at phase 2. What else is in their pipeline?
These are always so interesting to me. Like, how do we know what's causing schizophrenia? I'm very outside of the neuro psychosis space.
Yeah.
But I'm wondering here. Okay, so this is very interesting. So this is the actual asset in question. Okay.
Yeah.
Let's just like read through this because this word right here is what I was looking for. So Ariella scientists use crystallographic structures and pharmacological assessments to develop ART5803, the first precision therapeutic directly targeting the pathological effects of autoantibodies on the NMDA receptor. ART5803 is a humanized monovalent monoclonal antibody. That's the one arm. You can kind of see it there.
Yeah.
The FC region right here. Bing, bing, bing. Right there. And then can I zoom in here? Possibly. Oh, wow. Perfect.
Hey, let's go.
So here's the NMDA receptor here, right? Here's the single arm, the single variable region that's going to bind there. And then here's the FC. So see how much smaller it is without that extra arm? So it's much smaller. It might be better at passing the blood-brain barrier. But they say in preclinical models, ART5803 rapidly reversed behavioral symptoms caused by NMDAR autoantibody pathogenicity. Arialis has completed phase 1 SAD and MAD clinical assessments in healthy volunteers, blah, 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 blah. Okay, so there's not a whole lot of information on like how it actually works. I'm going to guess that they had to bind this thing in a very particular spot on the receptor to not block the receptor completely, but to block the pathological consequences of whatever's causing the disease.
Oh, right. Yes. It's not like you can just, put a sock over top of this thing and you're done.
Exactly.
You still need it to do its job.
Yeah. Yeah. So it's binding in such a way. This is probably what's happening. And if I win, I get a gold star. Here it is. It's probably binding in such a way that allows for the natural ligand to still bind to the receptor, but it blocks the autoantibody from binding.
Yeah.
And the autoantibody, when the autoantibody binds, the natural ligand can't bind.
Yeah.
So it's probably just like a small little blockade that allows the ligand to come in, but disallows the autoantibody from binding. That's got to be what it is. And you can kind of see it in their picture here. Like you can see if the ligand's going to come in and bind right here in this little pocket right here. And again, this is probably just this is not an actual like accurate depiction, but this right here is binding in such a way that allows the natural ligand to come in here probably, but it also blocks the autoantibodies, which are probably here in pink.
Yeah.
So yeah, this is what I'm seeing more and more and more in CNS stuff. Autoreactive, autoimmune CNS stuff. Very specific. Like this is very, very specific, like super precise.
Yeah.
Right. And this is just a consequence of just our better understanding of the diseases. Yes. But also our better ability to create these kinds of medicines. Really good.
Okay. Awesome. So yeah, it makes sense why this one has jumped to the top of the Biotech Voyager leaderboard. Really exciting. And again, given that this is novel, this is something that has no other treatment available. Really exciting to see. But yeah, to your point earlier, right there, they have another asset that's being developed for schizophrenia. I learned recently, no one who is blind has ever developed schizophrenia.
Really? There must be some kind of visual component to it. Yeah. Where'd you hear that?
I read it online somewhere, Jeff. I don't know.
Oh, it must be true then.
I think it was from a reputable source somewhere on LinkedIn, somewhere I trust. But yeah,
Nice.
just a little interesting fun fact, if you can call it that, for neuro degenerative diseases.
This is interesting too. This is a very interesting space. I hate using the word interesting so often, but this whole space, like the blood-brain barrier penetration and target specificity technology in autoreactive CNS disorders, is really interesting. I've got a few companies on the Biotech Voyager that I'd love to disclose, but I'm not going to, that are doing similar things.
Mm-hmm.
So yeah, keep your eye on that space. Arealis being one of them.
Right on. Excellent. Okay. Once again, folks, we're going to be having Ken Greenberg, CEO of SonoThera, on the show at 12 noon EST. If you have any questions for him, feel free to stick around and put them in the chat even now if you like. He's working on a really powerful method using microbubbles and ultrasound to deliver really large genetic payloads. Really exciting approach, kind of a third category. You kind of have the traditional non-viral LNPs, you have viral vectors, and this is kind of a brand new category for gene therapy delivery. So really looking forward to that. They just raised $125 million. And they've been on a little bit of a streak the last couple months in terms of releasing some pretty cool data. So looking forward to that. Okay, moving on here to immunophotonics, Jeff. You ready?
Yes.
All right.
So I have to admit, immunophotonics probably should be graduated from the program. They're phase 3. They're
phase
post.
3. Come on, Jeff. What's going on
phase 3.
here?
They're post Series B. But I like the company, and they posted on LinkedIn this morning about this milestone, and I wanted to talk about them because they're very cool. So I added the profile this morning, but I'm going to graduate it again later after the show. But anyways,
before we get into that, so like, what constitutes that? What constitutes a company graduating from the early stage biotech leaderboard on the Biotech Voyager?
Yeah. So, well, I'll explain what a company must, the stage at which a company must be in to be on the Voyager.
Okay.
And then that'll explain what causes them to be graduated. So I'm considering the Biotech Voyager to be tracking emerging biotech companies. Early stage, emerging, whatever. I use them synonymously.
Sure.
But I would consider a emerging biotech company to be Series A or below, and phase 2, early phase 2 or below. So I consider like Series B raises or phase 3 initiation or pivotal trial initiation. Sometimes you have phase 2 pivotal trial initiation to be graduation events. And it's just because I want to really focus on the emerging technologies.
Yeah.
So anything that if you were to walk into a crowd of people who are like biotech fluent and say, "Oh my gosh, here's this list of early stage companies," and you had like Novartis on it or like, Pfizer, it's like, okay, you've lost all credibility. Right. Yeah. So I had to put some kind of event to say, okay, this is probably relatively reasonable to say that this company is below or before this milestone. You could consider them to be still early stage and emerging. So yeah, pre-Series B and pre-phase 3.
Okay.
So immunophotonics being phase 3 and post Series B would qualify them to be graduated, but it's a cool technology that I want to talk about. So I made the profile just because. Okay, nice. There you go.
Okay, so Jeff, why are they one of your favorite companies? What's going on with immunophotonics?
Yeah, so well, this morning, their company page on LinkedIn posted that they had a patent granted for China. So big milestone. They can now probably use this thing in China and push it into clinical trials in China. But the reason that I like this company is because they are like they have developed a very cool innate immunology. Like, I don't want to call it an agonist. It's a one of a kind, like class of 1 therapeutic. And it has to do with like really what's the right word? Like nerdy, innate immunology stuff.
Okay.
So it's a cancer therapeutic. So we're going to start there. It's for cancer. It's for solid tumors specifically. Never can be used for hematological malignancies, because what it does is it's a infusion of a glycan polymer after you do solid tumor ablation.
Interesting.
That's all it is. Right?
So I'm
Now,
coming in, I've ablated my tumor, I've kind of like wrecked it out of there. I've, done whatever to it, and I'm putting this in, and what is it doing?
yes. So this picture does a decent job at it, I guess. So these little purple and pink things are supposed to be tumor particulates that are released after the explosion that is caused by ablation.
Right.
So if you don't know, tumor ablation is one of these kind of like crude, relatively like barbaric types of cancer therapy where you pretty much just like shove a catheter into the person's tumor and then you heat up the end of it and you just like thermoablate the tumor. And you could either do it very cold or very hot. Normally it's very hot, or some kind of like ultrasonic pulse or whatever. It's some kind of like ablation. And the easiest one to think of is just like zapping it with a big shock of heat, right? And what happens is you get thermal damage and just like tissue degradation from this thermal catheter.
Yeah.
And what happens is the tissue just kind of like melts, right? And the cancer cells explode and you get all kinds of release of cancer particulates. A proportion of those are immunostimulatory, cancer tumor specific, like antigens and damps and all kinds of like innate immunology stuff. Okay.
Yeah.
But the issue is that that environment, like post ablation, while those are kind of hanging around, you want the immune system to get in there and like catch those things and use them to train the immune system. It's just not a very good opportunity because it dissipates relatively quickly. The area is hot and not good for like cell function, right? So what Immunophotonics has developed is this glycan polymer that they put into the tumor after ablation, and it just acts as kind of like a net, and it catches the stuff, right? And simultaneously, it is very conducive to ingestion by APCs, like dendritic cells. So this glycan polymer net will catch a bunch of tumor particulates, and then it's taken up by APCs eventually. And the glycan polymer is immunostimulatory itself, and then the antigens are also immunostimulatory. So you get a very powerful innate immune response that leads to a adaptive response.
Gotcha.
Like anti-cancer T cells. And their big thing is they look for abscopal effect, which the problem with tumor ablation is if I have metastatic pancreatic cancer, God forbid, you could put a catheter, I guess, into my primary tumor and ablate that thing. But if I have a bunch of micromets all over my body, you can't ablate all of them and just like make me like needle face, right? So what you do is you ablate the primary tumor, put in IP001, Immunophotonics glycan polymer, and you get that adaptive response. And then your adaptive response against the tumor will go all over your body and you'll get an abscopal effect.
Right. Nifty. Okay.
Yeah.
That makes sense. I like it.
So, and they're in phase 3, I think, or at least phase 2-3. And they've been around for almost 20 years. The CEO, Lou, is super awesome. Go follow him on LinkedIn. Lou Azelero, Azelero. I don't know.
Nice.
Yeah.
Well, I think you can scroll down and you can see the company leadership, right?
Oh, true. Yeah, true. Oh my gosh, where's Lou? This is the wrong. I know Tom, Rob. Did they have a recent Oh, you know what? I think that Lou actually stepped down.
Oh.
Yeah.
Sorry,
not like
Lou.
in disgrace, but just
okay.
like yeah. Yeah, here he is. Let me
and then who's who invested in them? Who's been supporting them all this time?
Nice. Oh, they've got JJDCI. Oh yeah, see, he's now the director and advisor. Yeah, they're not like super capitalized, about 30 million. But I would imagine that this type of therapeutic is relatively cheap to produce. Right. All it is this like glycan polymer. So it's not like they're making some kind of crazy antibody with cell lines or a cell therapy or whatever. It's a relatively speaking, simple type of thing that you get some IP around that, and you're off to the races.
Nice. Would this be a good combination therapy? Is this something that would ever work well with a different solid tumor, like a CAR T therapy or something? Or is this something that you think will mostly just work with something like ablation, where you kind of you need that red hot skewer that goes in first and messes up the tumor, and then this just kind of acts as the net to keep everything?
Yeah, I think that they would need to have like this thermal ablation comes first.
Okay.
Right. However, I think that there is possibility of like a tri combination where you have the thermal ablation as like the first therapeutic, you can call it that, and then like a combination administration of the IP001 and like a checkpoint inhibitor, like an anti-PD-1 checkpoint, to ensure that the anti-cancer T cells are not going to be inhibited by PD-L1, PD-1.
Nice.
Which I would be surprised if they weren't pursuing something along those lines.
Right on. Okay.
Yeah.
Sweet. All right. I like it. Yeah, this is nifty, Jeff. Thank you for highlighting it. But yeah,
They're graduating.
No longer early stage, so we will never speak of it again. Eventually, we might occasionally talk about other companies, but we do want that are higher stage, but we do want to focus mostly on the early stage companies. All right. Yes. Speaking of early stage companies that are graduating, if you go back up for a sec, Jeff, SonoThera is going to be coming on in about 20 minutes or so. Ken Greenberg just raised $125 million to go after a brand new category of gene delivery, which is microbubbles combined with ultrasound for delivery. Really cool approach. Really looking forward to that conversation. If you have any questions for Ken, by all means, stick them in the chat, and we will get to them post haste. You can kind of have priority there. That makes it a lot more interesting than just me and Jeff jabbering at him. Okay, so yeah, SonoThera going to be on in about 20 minutes or so. So stick around for that. Don't go away. But moving on here, let's go on to the next one here, which is going to be Apligon. I hope I'm saying the name of this company correctly. So right off the bat, really interesting approach here. I think in the initial signal, Jeff, this one's older too. It's based in 2009. Does this one count as an early stage company still?
So they're probably pre-phase B, and they're still in phase 2. Okay. So they're getting close.
All right. They should be graduating soon, but yeah, pretty cool application here. So yeah, they're a Finnish company. I think this is the first Finnish company we've talked about on the show. I think there aren't a whole lot. Oh yeah, also you can go check out this really cool interactive map that showcases the different companies in Finland.
I'm going to show
which other ones can we see there? Can we click on it and see kind of like the list?
Oh, you know why it's doing this? Because I am on the tablet version. Yeah. So, Ben, help me out. Where's Finland?
Finland. I think you're in the right spot, or it might be there.
There we go.
I think Sweden's in the middle.
They're up here. Yeah. So, yeah, this is super cool. I mean, this map is really insane. I mean, you expect to get all the hubs: Boston, San Francisco, San Diego, and then all the EU hubs, the Israeli hubs, Asian hubs. Just showing all the locations of these emerging biotech companies. And this one I thought was a mistake. It's not. There actually is a company out here in the middle of the ocean.
What?
Yeah, in Bermuda. Or at least that's their stated location, is Hamilton, Bermuda. So
we need to get them on. We would be like, yeah, can we come interview you on location?
Yeah, on location. Yeah.
We will do a free Bio to Bedside episode.
Bio to Bedside, what's that?
Bio to Bedside. I am clearly off of it. Okay. Orion Pharma and Glykos. Are these ones that we should be looking at? Other ones? Orion Pharma, just in the comments here. Is Orion on there? Not yet. We don't have all of them mapped out. We're slowly building out that list, but thank you very much, Richard, who I assume is Acilia Therapeutics. Okay, but anyway, coming back to Apligon here. So they've raised 5.6 million euros, not to be confused with dollars. I was really confused for a second why it was E5.6M. The E is for euros. Apparently, the font kind doesn't have the euro symbol in it, but it's got the dollar sign. But yeah, really cool approach. So what they're going after here, the specific problem that they're targeting is when the surgical access point of dialysis patients fails to mature properly. And so it's kind of like an interesting blood thinner play, where essentially the problem with standard blood thinners is it just goes all over the body. And so, you've got, you need to thin the blood for whatever reason, someone's clotting way too easily or whatnot, especially around like a surgical point or whatnot. Or, a catheter point or whatnot, and it's kind of gumming everything up. And if you put someone on blood thinners, if it cannot be a great thing if they end up getting a cut somewhere else on their body, and they just bleed and bleed and bleed and bleed and bleed. And so Apligon in particular is developing a mechanism for a blood thinner that can be localized, which is really interesting, as opposed to something that's just kind of going systemically, from my understanding. So it's an interesting thing. I wouldn't have thought that you could ever make something that's specific to one area, but it's an anticoagulant that's localized.
Okay, help me out. Why would we want a localized anticoagulant?
My understanding, right, is in the context of this, it's usually for I saw a component of it here. Usually it's just with regards to the fact that you are like doing surgery, and you're not doing surgery, but like you're injecting a needle or whatnot, and basically you can get a bunch of clotting right around the needle, and it kind of prevents you from being able, it kind of like gums up the works. And so you do want a localized way to just make sure the blood flow keeps going through fine and everything. But you don't want that applied to the whole body. That was my understanding.
Okay, so it's saying here that they're doing it for like limb-threatening ischemia and like other kinds of arterial stuff, like thromboinflammatory vascular diseases, which makes sense, right? Like thromboinflammatory vascular diseases. I'm thinking, I don't want to sound stupid, even though I probably already do. Is like varicose veins something like that?
Maybe.
Or no?
Admittedly, it just kind of looked cool. I'm like, I've never heard of an anticoagulant that you can do locally. So I wanted to dive a little bit into the mechanism. So yeah, again, just what's interesting is you can just stick this into the injured spot, and it'll just act as an anticoagulant there. But
okay, so it does. This is kind of like varicose veins. You've seen varicose veins, right? Where people have like, it's gross. I'm not gonna pull a picture up, but you've seen them. A lot of people have them. They're like when your veins in your legs get all like bloated and like squiggly and
right and blue.
Yeah. So chronic pooling and inflammation damage the endothelium of the blood vessels, altering the balance of coagulation. It's a systemic and localized inflammatory state, and it increases the risk of blood clots forming. Oh, duh, blood clots. You would want to like disrupt the formation of blood clots.
Yeah, and I guess you can do it locally, like in the legs. I guess.
Yeah, that makes way more sense.
I misunderstood.
A systemic blood thinner. Yeah, that's cool.
Like, for example, my grandmother, sadly she has passed. She was on blood thinners like all of her life, like for the last like 10, 15 years of her life, because she had that problem. She would sit down for like extended periods of time, and like blood clots would form in her legs, and she like almost died a couple times from like those blood clots then, breaking off inside of her leg and going up to her brain or whatnot. And so they just kind of had to have her on permanent blood thinners. But potentially, if I'm understanding correctly, with something like Apligon, this would allow you to just administer it locally to that area, so you don't have the blood clots forming.
Oh, okay.
Yeah.
I'm hesitant to pull a picture up here, but this is a relatively innocuous one. Here we go. This is what it's supposed to look like.
Okay. Yeah.
You've seen those. Okay. That's enough.
my grandma had those.
Yes.
Yeah. Cool.
Interesting.
All right. So yeah, sadly it won't benefit her, but she's in a much better place. So we can we can move on from this story. But yeah, interesting to see. Yeah, it's I believe the first Finnish company, but yeah, if you go to the Biotech Voyager, you can check out all the other companies that are in Finland as well that are early stage. And then folks, if at any point whatsoever, so maybe that's why Richard has put these two different companies in here. Maybe we're missing these, Jeff. Glykos and
Orion is definitely
Pharma.
not early stage. He was probably answering the question of what else is in Finland.
Oh, okay. Gotcha.
Yeah.
Cool.
And then Glykos, I'm sure Glykos, yeah, like GSK, is that GSK? Oh no, never mind.
Yeah.
Yeah, Finland. Okay. He's just talking about Finland companies.
Nice.
Yeah.
Cool. All right. Jeff, ANACA, one of your other favorite companies, correct?
Yes.
Do you want to take a look
So
at that?
in fact, this
Glykos
Glykos should be on here. I'll get this one on here later. Yeah, so ANACA. Let's talk about it.
All right, sweet.
Very cool company out of Sweden. They are a TCR-T cell company, and they just recently dosed their first patient.
Hey, I believe that deserves an applause.
It does indeed.
If I can flip and find it. Here we go. Yeah.
Congratulations.
Good job.
So they have a very interesting approach to TCR discovery. So everyone knows CAR T cells, right?
Yes, they do.
This would be a good time to talk about the differences between CAR T cells and TCR T cells. Because we haven't had a TCR T cell company on here yet, have we?
I don't believe so.
Yeah. So they're
my love.
Company, not a CAR T cell company. And what TCR T cell companies do is they create synthetic TCRs, T cell receptors, right? Kind of. And basically the pitch is that CAR T cells have the intracellular domains that activate the T cell. The extracellular domain is like an antibody that binds to a target, right? But that target has to be a extracellular or surface-bound molecule like CD19 or HER2 or whatever have you, right? But it has to be protruding from the cell surface to be bound by that antibody. That limits a lot of the cancer-associated antigens because a lot of cancer-associated antigens are mutated intracellular peptides, proteins. Right. How do you target like a mutant KRAS with a CAR T cell? You can't, right? With a CAR T cell. You can't with a TCR T cell because TCR T cells mimic TCR biology, and TCRs bind to peptide-loaded MHC complexes, and you can have mutated peptides, like a mutated KRAS, loaded into MHC, and then that can be recognized by a TCR T cell and a normal T cell. So what ANACA does is they have created a TCR T cell that recognizes a particular HLA subset that is loaded with mutant KRAS, G12V KRAS.
Okay.
Which is very important or very implicated in pancreatic ductal adenocarcinoma, PDAC.
Right.
One of the most common forms of pancreatic cancer. So this is a cell therapy for pancreatic cancer.
Nice.
Which is really insane. Yeah. So, but they're, and they dosed their first patient. They dosed their first PDAC patient. I'm thinking that this is probably happening in Sweden. Yeah. Yeah. So I'm, not that it matters, but here, yeah, G12 and G12V and G12D mutations in KRAS affect 90% of pancreatic cancer patients, blah, 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 blah. VIDAR addresses this unmet need by engineering the immune system's T cells to recognize and attack cancer cells carrying the KRAS mutation. So what's really interesting about ANACA is the way that they discover the TCRs. Okay. Because you need to be able to generate a TCR, right? Like a CAR T cell is just an antibody fragment. And you can pretty much just like use the formulation of like a CD19 targeting antibody as your binding domain. But to create a TCR that binds to like HLA-O2 bound mutant KRAS is very complex, very complicated. And I won't go into all the details, but they have a dendritic cell-based screening platform that they use to discover potent and specific TCRs. It's really, really cool. The company's awesome. The guys who run the company are super cool. And fun fact, we're going to have Regan Jarvis, the CEO, on the show next Tuesday. And this thing is
let's
ruining my RAM right now. Their website is insane. They probably spent 30 or 40K on this website. But it's really cool. We can watch this movie. You can go watch it yourself. They have or at least an antigen presenting cell based screening platform to discover TCRs. Really, really cool stuff. And I'm a huge cell therapy nerd, huge T cell nerd, and I love to see cell therapies for solid tumors, and especially for things like pancreatic cancer. Huge.
Cool. Interesting. Yeah.
ANACA.
And again, Jeff, why is it that obviously pancreatic cancer is such a big deal here?
Well, we talked about it with the guy from the GLP-1 gene therapy thing. Who was that?
Oh, Jay Kaplan.
From
Fractal Health.
Fractal, yeah. So we discussed with him, the pancreas is just hard to get to.
Yeah.
And they're doing their GLP-1 delivery to the pancreas. They do it like directly to the pancreas through like the esophagus and like stomach route. It's crazy. To get to the pancreas, it's just really hard. So treating it with like surgery and stuff has just been notoriously challenging. But also pancreatic cancer is two more things. Relatively asymptomatic until late stage, which is not good because you have no idea you have it until it's like everywhere in stage 3 or 4. And then 2, pancreatic cancer is notoriously like it creates these like super thick fibrous networks, and it's very hard to get like the immune cells in, drugs in, so it's hard to treat.
Okay.
Yeah.
And again, it's also like one of the most deadly cancers on the planet as well. Just separate from
for those reasons. Like Alex Trebek had it, the guy from Jeopardy, and they don't find it until it's super late stage because there's just like no way to know. I mean, and there's probably, I don't know this for a fact, but there's probably not a whole lot of good screening methods to diagnose early. Once you get diagnosed, it's usually because of pancreatic cancer is normally diagnosed because of symptoms, getting symptoms from metastatic disease. So it's like, I've got this thing on my rib cage, this thing and they look into it and it's like, oh, this is a metastatic pancreatic cancer, right? So that's the problem.
Yeah.
So
yeah. And so again, is this like the first cell therapy in the world then for pancreatic cancer, or are there other ones going around in that area? You don't have to answer off the top of your head.
Yeah, it might. That would be really cool though if it was like the very first cell therapy, like the first patient dosed for a cell therapy for pancreatic cancer in the world. Possibly, that's possible.
Okay. Cool. It's it's so again, if we're just kind of going back for a moment here, Jeff, and we're kind of again just looking at the difference here between like a standard TCR-T cell and a CAR T cell. Again, like what's the difference here between like a TCR and a CAR, a TCR and a chimeric antigen receptor?
Yeah, so a TCR binds to the canonical like HLA peptide complex that all of our cells present to the immune system. When you get a viral infection, the virus will go into a cell, create a bunch of itself. Part of those proteins from the virus are processed by the cell itself and presented to the immune system. And the immune system can see that, the T cells can see that peptide in the MHC complex through their TCR. A CAR is like a Frankenstein kind of version of that. It's very, very Frankenstein, where you have like a couple of the T cell receptor, like sort of activation domains shoved in conjunction with an antibody domain. And the antibody will bind to a target and then kind of mimic TCR signaling, very rudimentary. That's the biological difference. Therapeutically, the difference really is that CARs are restricted to surface antigens, whereas TCRs can bind to any foreign protein.
Right. Interesting. Okay, so yeah, it's very much the difference between something that's highly specific and something that's a bit more generalized with something like a TCR.
I'd say TCRs in theory have more of a targetable repertoire, whereas CARs are very restricted. But the thing is that it's very difficult to create the TCRs themselves, which is why ANACA's approach is so interesting, using biology itself, dendritic cells, as a screening platform.
Interesting.
Yeah.
Okay, right on. Well, we've got 5 minutes here before Ken comes on the show, so maybe we can just quickly go on to the next story here before he comes on. Edgewise Therapeutics. How does that sound?
Sure.
Excellent. Okay. Edgewise Therapeutics. Where do they sit on this whole thing?
They're 28. So if we do like a rare disease search here, let's go to the leaderboard here. Leaderboard. Filters, rare diseases. Let's see, whereabouts are they? They should be up here somewhere. What was their score? Oh no.
Edgewise. Oh yeah, I see them. Go back down. You missed them.
Did I miss it?
Like number 8 or 9.
Oh yeah, okay, it's just cut off. Okay, yeah, they're there. Bing.
Right on. Okay. So yeah, if we cut through here, what's going on? So again, this is a heart drug, and this one's interesting because they're focusing on like the myosin, like the actual cytoskeleton here, and I was a little bit confused when I was first looking at this one, because you'd think with a heart disease maybe you'd want to strengthen the heart a little bit. And so my simple brain is like, okay, well then I want to strengthen myosin. I want to increase it. I want to increase the cytoskeleton to some extent, right? To bolster up the cells. It's actually the opposite. Basically, these heart cells thicken up way too much. And by thickening up way too much, you actually cause all sorts of other problems. And so it's kind of a little bit counterintuitive. This therapeutic is designed to actually disrupt the myosin. It's designed to basically reduce the thickening, which I wouldn't have expected, right? Whenever you think of, muscles, you think, well, I want to increase my myosin filaments. I want to work out. I want to make my muscles bigger. But there's kind of like this, I guess the heart gets too muscle bound in this circumstance. And so you'd actually need to scale that back. Sorry, bodybuilders.
Yeah. So from my understanding, it's what happens in these diseases is the heart is like contracting too hard, too much, and you have to relax it a little bit. Let the heart fill, and then it can. Yeah. So I'm not super familiar with like cardiology really, but I am relatively familiar with like the actin and myosin. Or oh my gosh, here I go. The actin and myosin interactions.
Right.
I did a lot of stuff in like my undergrad with like actin and myosin and like fast and slow twitch muscle fibers and stuff. So it's a very interesting like protein dynamic happening in our muscle cells.
Yeah.
But in terms of the actual like how this is working, I'm not going to even pretend what sarcomere modulators do.
Well, basically, from the looks of it, it looks as if basically you want to still keep the squeeze of the heart high, right? You want to actually keep the blood pressure at that reasonable rate. And I guess a lot of other drugs inside of the space that are trying to deal with this particular disease, they relax the heart, but they relax it so much that the squeeze itself gets reduced as well. And I guess they've kind of been able to thread the needle here a little bit, where essentially the therapeutic approach they're taking here allows the squeeze to stay really high, but basically just allows you to relax a lot more. So it's kind of like a whole like work hard, rest hard situation, where you still are able to squeeze, you're still able to keep the blood pressure at that acceptable rate, but you're also in between the squeeze allowed to really relax, and that helps with heart longevity and that sort of thing. So yeah, it seems that across like hundreds of heart scans, there's no penalty to pumping strength here. So the different people who have been in it so far have benefited. They haven't had this other problem that other drugs inside of this particular indication have struggled with. And so, yeah, it's kind of opened up a new category for people with this hypertrophic cardiomyopathy. So, yeah, it's pretty nifty. I was quite impressed to see this, and I know like it's a little bit earlier or further down the list, but still super interesting approach regardless.
Yeah.
And so where is this trial taking place? Can you click through the press release there?
Let's see. They normally have like a about this thing. Look for a company or look for a. Well, they're located in Colorado, so it's probably U.S. based.
Nice.
So.
Well, yeah,
it's interesting. So, was this the last one on our list today?
I think we also have Nucellix we could go through quickly.
Nucellix. Did you pick that one?
I did.
My computer is losing it.
That's fine. Okay.
How do you spell that? Oh, this one. Okay.
Yeah. Okay. Ken should be joining soon. Let's see here. I'm going to just send him a text. Make sure.
MS.
Okay.
Whoa. Okay. This is crazy. What? Wait a minute. Huh.
Nice.
Okay, cool. Yeah. Wow. Okay. It's an embryonic stem cell derived astrocyte therapy for multiple sclerosis. This is what I'm talking about, dude. Regenerating neurons, even though astrocytes aren't really neurons, but like stem cell based regenerative medicine for CNS disorders. I love it. To treat chronic demyelinating diseases, including multiple sclerosis.
Gotcha.
AstroRx, yeah. So here's the paper in stem cell. Pro-repair properties of human embryonic stem cell derived astrocyte cell therapy and demyelinating disorder. Wow. That's really cool.
Yeah.
It enhanced microglial phagocytosis of myosin debris, reducing inflammation. It extended lymphocyte proliferation and survival without excessive activation. That's good.
Yeah.
Enhanced murine. Okay. Huh. Intraventricular delivered AstroRx promoted microglial mediated myelin debris clearance and oligodendrogenesis. Oh, wow.
Yeah.
Okay.
No, it's nifty. Anyway, we could revisit this in just a little bit because Ken has joined the studio. Unfortunately, I was the one who was the failure here.
Oh no!
When I sent over the calendar block, it also included a Google Meet link. So we're we're good to go now. Okay, so ladies and gentlemen, very excited to have Ken on the show. Without any further ado, let's just bring him in.
Hey there. How are you?
Good to see you.
Good to see you too. Thanks for having me.
Oh, more than happy to. We appreciate your time. So yeah, Ken, like take us through it. You just raised another tranche of money. Can you tell us what happened there?
So we just closed our Series B financing. We raised $125 million from a great syndicate of both venture and strategic investors. Vita Ventures led the round, got participation by a number of really top-tier venture and strategic groups, as well as our existing insiders also participated. So we're very grateful and excited to have funding to move our platform forward into the clinic. So, over the next year, we're planning to enter into our phase 1 trial for Duchenne's with our lead program. And then we've also got a program in ADPKD that we're advancing behind that, as well as additional platform and pipeline opportunities that we're pursuing. So it's really exciting times here at SonoThera.
Excellent. Well, I sent you over the link this morning, Ken, but we had one little clip in particular on Instagram that blew up, really just kind of overviewing your approach. Hopefully, we did justice to the approach that you're taking. But it's really exciting because it is kind of opening up a brand new category, right? When people think of gene therapy, you kind of think of one of two approaches, one which is non-viral, LNP based, maybe some different nanoparticles, versus, and then the other side being viruses. And you guys have really pioneered like a brand new category here. And so would just be really curious to get your overview once again of how this particular approach works.
Sure. Yeah. So basically the entire idea is to, as you mentioned, avoid the use of viruses, avoid the use of any type of a carrier whatsoever, which can be immunogenic. And it kind of funnels you down this path of making a number of compromises, right? If it's a virus, you can't redose it. You're limited by payload capacity. It doesn't necessarily target to the organ that you want it to go to. The manufacturing is really complex. So our approach is really to remove the virus entirely. And the way it works is to take a naked genetic payload. It can be DNA, RNA, et cetera, gene editors, ASOs, siRNAs. It really doesn't matter what the genetic payload is. And we co-infuse that with a contrast agent that's used for ultrasound. These are typically referred to as microbubbles. They're composed of either a lipid or a protein shell, and they contain an inert gas core. And they've been used safely for decades for diagnostic contrast-enhanced imaging. So we pair those two components together, inject them IV, and then they're flowing throughout the body in circulation. However, only where we apply the ultrasound energy do we enable delivery into that target organ. So it's highly targeted by virtue of where the sound waves are actually directed in the body. Now, when the sound waves activate the microbubbles, there's several mechanisms that actually mediate the delivery, first to escape from the vascular system into the target organ. Second, to create these transient pores that form in the cell membrane that enable intracellular access. And then third, to modulate the nuclear pore itself so that we have entry of the payload into the nucleus for transcription and downstream translation to occur. So there's multiple steps along the way. They're all mediated by the ultrasound and the microbubbles themselves. And it's highly controllable without all the issues that arise from using viruses.
So forgive my ignorance. Duchenne's muscular dystrophy, where would you apply the ultrasound energy for DMD?
So DMD affects the entire skeletal muscle system, as well as the diaphragm and the heart. So typically patients die from cardiopulmonary failure. So for our DMD program, we're actually applying the acoustic energy throughout the muscular system. So, we're using a handheld ultrasound probe that can be physically rastered over the muscle. So we cover the entire body. Initially, we'll be treating the skeletal muscle, and then in more advanced patients, we'll go into the heart and the diaphragm as well.
So it's in effect like a systemic delivery, or can you
Exactly.
control muscular delivery somehow?
Well, the payload and the microbubbles are administered systemically.
Yeah.
And then we apply the acoustic energy to the muscle groups that we want to treat. But we do target it. So, it's targeted by virtue of where the energy is applied, but also within our genetic payloads, we incorporate muscle-specific promoters. So we've got the regulatory elements that will drive gene expression only in muscle fibers without having off-target expression in other adjacent cells.
Right. Would there be toxicities of dystrophin expression in like other tissues?
I'm not aware that that's been reported, but we always want to try to recapitulate the endogenous gene expression as much as possible. So, there's always a chance that if you were to express ectopic dystrophin in immune cells, for example, like that could lead to, adverse events or immunogenicity, which we just want to avoid any possibility of that at all costs. So we do drive expression with muscle-specific promoters.
Nice.
So it's sorry, one more thing, Ben. Sorry. So it's possible to control like the depth within the tissue that you can penetrate, like with some kind of. I'm not a biophysicist by any means. So
it is, and that's done clinically all the time for diagnostic ultrasound. So you can think of it, there's this beam. It can be, parallel or it can be focused. And if it's focused, you can adjust the focal depth of where that beam actually hits its apex within the tissue. So that's an easy adjustment that can be done bedside to adjust the focal depth, essentially, of the energy. And we do that all the time because you want to make sure that you're imaging beyond more superficial tissues like the skin or adipose or even bone. You can image through all of these tissues by adjusting the focal depth.
Nice.
So I guess I have one more question then. We were talking to a company called Fractal Health the other day, and they're doing a pancreas local delivery of a GLP-1 gene therapy. Would it be possible to use this approach as a less invasive and more effective delivery to the pancreas?
It would be possible. There have been preclinical studies using ultrasound and microbubbles to deliver various genes to the pancreas. So I think that is very much feasible. Anytime you can avoid an invasive route of administration, I think there's a preference for doing that, not only for physicians, but patients and risks of complications and things of that sort. So our approach for all of the organs that we're going after is really to maintain as a non-invasive route of administration as we possibly can. So it's all IV based, and then we get essentially the local targeting by virtue of where the energy is applied.
Cool. All right. So I just learned something new in this conversation that I would love to dive a little bit more into. So my understanding up until this point, was that you were able to basically open up holes in the outside of the cells, right? Like through the plasma membrane, you can get into the cytosol. It is new information to me with regards to being able to then deliver, to also influence the nuclear, the nucleus or nuclear delivery as well. So how exactly does that work? Is it kind of the same concept that like the ultrasound kind of vibrates and creates some holes in the nucleus and allows entry that way, or is there something else that's secret sauce?
Yeah, so the nuclear entry has been a huge hurdle for non-viral gene delivery. If you look at LNP mediated delivery, you can, depending on the cells, especially in the liver, you can get reasonable gene delivery across the cytosol through receptor-mediated endocytosis, but getting across the nuclear envelope has been a major hurdle. And typically, publications that show nuclear entry with an LNP, it's around 5% or less. It's single-digit percentage of all the DNA that's delivered. The fraction that gets into the nucleus is very low. So with our technology, we modulate the nuclear pore. So we're not creating new pores within the nucleus, because the microbubble actually never enters the cell, right? The microbubble,
Yeah.
it's collapsed extracellularly, it creates the pores. So the the nuclear entry occurs by virtue of the ultrasound itself. And the main mediator here is calcium. So when you create these pores, there's an influx of calcium through the pores. So you have this transient elevation of calcium in the cell. There's also a release of calcium from the ER. So from the endoplasmic reticulum in the cell, you have an additional release of calcium that occurs from the acoustic energy. So these two things basically work in concert. You have a transient elevation of calcium, and that causes the nuclear pore to remain open longer. So it increases the open state kinetics of the nuclear pore, allowing for a greater amount of DNA to passively diffuse through the nucleus. So that's one mechanism that we leverage. It's this passive diffusion because the door is essentially open longer.
Right.
But within our payloads, we also incorporate DNA targeting sequences. These are sequences that basically bind to transcription factors. So they allow for shuttling of transcription factors across the nuclear envelope. So it's more of an active transport process.
Okay.
So we pair those two together to get very efficient nuclear entry of our payloads, exceeding 50%, which is really unheard of in the non-viral world. And it's really the key to being able to actually drive high levels of efficient gene expression.
So you're looking at like a 10-fold increase. Kind of the standard right now inside of the field so far with non-viral has been like 5% max, and you guys are at 50.
Yeah. It's
Nice.
a very efficient process, and this is something that, we didn't discover this. This has been published in the academic literature around the effect of ultrasound energy on the nuclear pore complex and how it modulates the open state kinetics by virtue of this calcium mechanism.
So then I have to ask, why you and why now?
We get this question a lot. So there has been a rich history of sonoporation in the academic world. You can see, dating back many years, the application of ultrasound and microbubbles in a variety of different animal models. So, why now, why us? So we started the company back in 2022 because myself and my co-founders, we really thought that there could be a better way to deliver genetic payloads and avoid all the issues that we've talked about. So we came together and we brought essentially my background in gene therapy and genetic medicine more broadly. My co-founder and CSO, his name is Steve Feinstein. He actually invented the first two FDA-approved ultrasound contrast agents. He's a clinical cardiologist, and he was scratching his head wondering, why is there not a contrast agent for ultrasound, which is really a core modality for imaging in the heart, but there's a contrast agent for every other imaging modality, MR, CT, and PET, right? So he invented the microbubbles back in the 90s, but then he discovered that they actually serve this additional purpose of mediating the delivery of therapeutic agents. So fast forward to 2021, we came together and decided, let's really try and take this core technology and translate it into the clinic. We felt that the time was right because the scientific and the investment community, I think, recognize that the limitations that are inherent in AAV they're just inherent in the modality itself, and it's been very challenging to work around those. And the progress with LNPs and retargeting them had not progressed as much as we had all hoped either. So we came together, and it turns out it's actually quite complex and costly to bring together the right expertise across ultrasound and acoustic engineering and microbubbles, and gene therapy. Like, typically these fields don't really converge all that much. So, one big thing to overcome was really just integration and bringing together this multidisciplinary team. Now, for the first year and a half or so of the company, we used basically vanilla sonoporation approaches, and it worked reasonably well in rodent models. But when we tried to translate it to large animal models, and especially non-human primates, it became clear that there was a real limitation in achieving the efficiencies of delivery that we needed in order to move forward into the clinic. So this was really, it was existential for us, because we had to really overcome a limit in terms of the efficiency and the depth of delivery and the biodistribution that we could achieve. So Steve, being an incredibly creative scientist, he really went back to the drawing board and reinvented how we can deliver the energy. And I'm not going to get into all the acoustic physics of it, but it basically allowed for us to achieve about 100-fold higher levels of gene delivery efficiency across every single organ that we tried. First, we did liver, we then did kidney, then muscle and heart. It works across the board. So this was a eureka moment for us. We named it Ripple, which stands for remote induction of pulsed pressure lateral to energy.
Well done. The US government would pay like $50 million to make an acronym like that.
Yeah. So, Ripple is really the secret sauce that when we kind of look at the field of sonoporation, we think that this innovation has allowed for us to achieve a level of gene delivery efficiency that's really ready for clinical use and scalable to large animals and to humans.
So it isn't the traditional like sonoporation that's been used for decades. It's a brand new like type of biophysics. Is it safe? Is there problems with safety or concerns with safety?
Yeah, so it is extremely safe. We've looked at all of the, traditional biochemistry markers, whether it's in liver or kidney or muscle. We've looked at immunogenicity markers, signs of cellular stress or apoptosis. It is incredibly benign. It appears to be even safer than traditional sonoporation, because in sonoporation, you're basically applying energy, which is cavitating the microbubbles, which is essentially they're rapidly expanding and shrinking, and that can lead to endothelial cell stress. There's local heating effects, and you have to use quite a lot of energy in order to get the delivery. With Ripple, it's a fundamentally different delivery of the energy. So the pressure wave in Ripple, so basically the way Ripple works, it's kind of like an earthquake that triggers a tsunami wave that can travel throughout the ocean over, many, many miles, right? Ripple's very similar. So we apply this focused beam of energy, it hits the target organ, and it triggers a secondary pressure wave that travels throughout the vasculature in the target organ. And that pressure wave is actually what drives the microbubble collapse and all the mechanisms that I talked about at the outset around creating pores in the membranes and things like that. So this use of a second pressure wave is actually allows for much, much higher levels of efficiency of delivery and more uniform biodistribution in these three-dimensional organs, rather, which we couldn't get with sonoporation. It was with sonoporation, you literally have to cover the entire organ with the transducer, and there's acoustic shadowing and all sorts of things. With Ripple, it avoids all of that because it's basically a focal point, and then the propagation occurs throughout the organ.
Right. It's like splash damage in a RPG game.
Yeah, exactly.
Yeah.
Good analogy.
Yeah. That's
Right
very
on.
interesting. And again, like, I'm not a biophysicist, but this kind of stuff, we had a bunch of like acoustic acoustic therapy mediated stuff in my graduate program that I was like, that's for nerds. I'm not doing any of that stuff. But here we are in the future.
Definitely. I mean, I heard about this when I was in grad school, like traditional sonoporation, and we were using pretty much every flavor of virus you could think of in our lab. And we were also doing biolistics, right? Gene guns, shooting DNA into tissue. And I had heard about sonoporation, but like you, I was like, that just seems like from another planet. Like, that's out there, right? But here we are.
Yeah. I think the other thing that's really cool here too, that I hadn't really realized, Ken, until we chatted at ASGCT, the challenge with kind of the two standard gene therapy modalities when we think of either LNPs or AAV, right, is any time that you make a new one, you've kind of got to go through the whole regulatory rigor of making sure that it's safe once again. While you guys kind of benefit from the fact, right, that you're using these microbubbles that have already been around for a really long time. Is that fair to say?
Yeah, so that was part of our strategy from day one. We wanted to try to minimize the regulatory hurdles as much as possible and use the FDA-approved microbubbles as well as the FDA-cleared ultrasound hardware. We modify the software in a way to deliver our Ripple acoustic profiles, but all of the existing ultrasound components, like you say, they've been around for decades, and there's quite a lot of safety data behind them. So it's interesting in that regard because it's very versatile as you think about AAV and LNPs. If you want to target a new organ, you have to develop a bespoke capsid or a bespoke composition of lipid and target the liver, the spleen, and retarget the new organ. Like that's extremely challenging to do. And then, as you mentioned, like you've got a new composition there, and it's got its own risk profile that needs to be de-risked. So in our case, we're simply redirecting the energy to a new target organ in order to enable the uptake. Everything else remains the same.
Super cool. So how does that work then? So basically the targeting component is primarily driven by the promoters that you're putting inside of the gene of interest, for example?
Right. Well, there's organ level targeting by virtue of the acoustic energy, and then there's cell specific targeting by virtue of the promoters and enhancers and other genetic regulatory elements that we include. If we use a ubiquitous promoter, we basically drive expression in pretty much all the cell types in the target organ at their endogenous distribution profile. So the energy and the microbubbles, they don't seem to have like a natural tropism. We kind of get into everything, but we are very selective in where we actually express the genetic payload.
Can they cross the blood-brain barrier?
That was my next question.
Oh, wow. You guys are on the same wavelength. They can. So we've done work in the CNS where we've disrupted the BBB and delivered both DNA payloads and siRNA knockdown payloads. There's actually a few clinical studies have shown that ultrasound and microbubbles are both safe and effective for delivery of other therapeutic agents into the human brain. So this has been done with aducanumab for treating Alzheimer's, as well as various chemo agents for treating glioblastoma. So a common question is, can the energy actually pass through the skull? And the answer is yes, absolutely. The key is to use a slightly lower frequency that can easily penetrate through the brain, through the skull and into the brain.
And can you do non-nucleic acid payloads, like maybe nanobodies or small molecules?
You can. So, the study that I just mentioned in glioblastoma, so that was using small molecule chemo agents. There have also been clinical studies using gemcitabine, so another chemo agent for treating pancreatic cancer with ultrasound and microbubbles. So it's not limited to genetic payloads. It's been done with antibodies and proteins as well. Our focus at SonoThera is in genetic payloads across the board. So DNA, RNA, gene editors, ASOs, siRNAs, but we're not working outside of the genetic medicine space.
Okay.
Nice.
But does your IP protect that? Because could you move into that space eventually?
Or if you can't say, that's okay.
we potentially could. Yeah.
Nifty.
That's not our focus right now, though. We're pretty busy.
Yeah, that's fine.
There's a lot of other things to keep you occupied.
One at a time, I guess.
Yeah. Awesome. Well, Ken, we want to be super respectful of your time. We know you're a busy man. We really appreciate your time. There is one question we're just kind of we ask each of our guests here at the end, which is basically what's being slept on in biotech right now that's going to be a big deal in a couple of years.
A big deal in a couple of years. I'm very optimistic. If you think about, I'm going to focus on gene therapy and gene editing. There's been a lot of progress in gene editing in particular. I think the holy grail is to be able to deliver a functional piece of DNA in a highly targeted way into the genome that can replace a mutant gene and integrate and drive expression by the endogenous regulatory elements, all the promoters and enhancers that are present, so that you faithfully recapitulate the natural biology. I'm super encouraged. I think that that's a really exciting emerging area that, quite honestly, has been a little stagnant because of delivery. And I think if we can pair, really excellent delivery with new editing capabilities, that's going to just open up a lot of diseases and indications that have up to this point been really, really challenging. So that's something I'm personally very excited about.
Fractal Health mentioned that last time. They have a, I forget the actual responsive element, but it's a promoter that's responsive to, like, this is going to sound stupid, but like eating, and they'll get their insulin expression when the person eats, but it's based on a very simple promoter-based regulatory element. You're talking about the actual endogenous regulatory elements, including enhancers, promoters, all those different things. Can you give me one example of where you think that would actually create a significant therapeutic benefit? I'm just curious to know your thoughts on that. Yeah.
So, there are certain diseases like Rett syndrome, as an example, where it's a bit of a Goldilocks situation. You need, if you express too much of the gene, it can be toxic, not enough, and you don't see a therapeutic effect. So, that's a case where when you bring in these exogenous promoters, you can easily overshoot it or undershoot it. And by essentially knocking in the wild type copy of the gene and harness the endogenous regulatory elements, you completely remove that risk. You're going to drive the native levels in the appropriate cell types, and the distribution will be as natural as possible. So things like that, I think, are really in need of better spatial and temporal regulation profiles.
Yeah, that was the initial promise of CRISPR technology. It was like the super precise, you can have these massive genomic cuts and get like the homogeny to get like the endogenous insertions. Hasn't panned out as we expected it, therapeutically, at least recently.
Yeah, I mean, I think CRISPR, it's still a very, very, in the grand scheme of things, new field, and it's still emerging. And it's also been limited by delivery modalities.
Exactly.
And, AAV and LNPs, they have certain use cases where they're showing promise, but a lot of the editing companies are focused on the same targets in the liver with LNPs, so I think delivery has also hampered the more widespread application of CRISPR, but the actual editing tools have really, evolved quite dramatically over the last few years.
Yeah.
So, yeah.
Definitely. Well, once again, Ken, thank you so much for your time. We know you're a busy man. Really appreciate you coming on, and congrats once again with the raise. Really excited to see the data you guys are going to be able to generate and release over the coming months and years.
Thank you. Appreciate it. All right. Well,
excellent.
Take
Thanks,
care.
Ken.
All right.
Thanks, Ken. Cheers.
Cheers.
All
Bye-bye.
right. Thank you for coming. Awesome. Oh, great guy. Really appreciate him coming on. This is awesome. And actually the other thing I was thinking about just at the end there too, Jeff, with regards to like how immature gene editing is, right, in CRISPR. I think Jennifer Doudna got the Nobel Prize in like 2012.
Okay.
So
somewhere around there. Yeah.
That's like the age of grade 8, grade 9 kid.
Right. it's in middle school.
Yeah. So
yeah.
Makes sense. CRISPR needs to grow up a little bit, but we'll get there, and it'll be a nice human being by the time it hits its mid-20s, maybe.
Mm-hmm.
Excellent. Okay. So last story of the day here. Let me just change over the banner. Edgewise Therapeutics. We had a chance to just talk about them a little bit before Ken came on. So maybe we can finish off with them. Really interesting approach here. Oh wait, was it Edgewise? No,
yeah, it wasn't Edgewise.
Sorry, here we go. Nucleus. Yeah, we had a chance to talk about them already. Okay, yeah. So an astrocyte cell therapy. So right, when you kind of think of a cell therapy, you very rarely hear about astrocytes. Jeff, would you care to elaborate a little bit on what astrocytes are, if you've got a definition off the top of your head?
So I don't really know a lot about astrocytes. I'm more of a microglia guy. They're a CNS specific cell type that exists mainly in the brain.
Kind of support it. They're like a supporting role cell type inside of the brain.
Yeah, I think that they help microglia. Hang on, real quick here. Yeah, the way that I know them is like kind of being the helper to microglia. But I would be lying if I pretended to know exactly what these things do. Right. But it seems like from the paper that this signal right here was, they published some preclinical data in cell stem cell, and in this paper abstract, at least, they're talking about how their AstroRx enhanced microglial phagocytosis. So microglia are like the brain resident phagocytes, like macrophages, but in the brain. Right. And they can consume and digest myelin debris, which will reduce inflammation. They extended lymphocyte proliferation and survival without excessive activation, which is really, really good. The excessive activation of the lymphocytes would actually lead to inflammation. And then enhanced murine and embryonic stem cell derived OPC differentiation. So basically what's happening here is their astrocyte, I'm wondering what the actual therapeutic rationale is here. If they're just basically saying like, hey, astrocytes can help here, let's put more astrocytes in. Or if there's specific subsets of diseases that's characterized by lack of astrocytes or impaired astrocyte activity. So, but their thesis is if we can get more astrocytes in there, they will relieve the symptoms of things like MS.
Mm-hmm. Yeah.
So
MS is such a big disease too. Like we were already talking about a neurodegenerative disease earlier, kind of on the opposite side. So yeah, it's so nasty.
But they're also doing some type 1 diabetes stuff with creating functional islet cells that secrete insulin.
Are they?
So they just seem to have a human embryonic stem cell platform that is pumping out all kinds of different functional cells, like astrocytes and islet cells.
New Celix. Right on. For the win.
Yeah, I guess the company name, like New Cell, right?
Right.
We're creating new cells. So pretty cool.
Sweet. Excellent. Well, Jeff, I think we can leave it there. We've been able to cover some pretty incredible companies today. We were able to go through some really interesting aspects of applications for neurodegenerative diseases. We were able to take a look at Aerialis, just kind of jump to the absolute top there with their phase 2. We were able to take a look at immunophotonics, a really interesting, almost like a polymer to help with ablation of tumors, to kind of keep everything contained while you're in the middle of skewering it with a hot poker. We were able to take a look at Apligon and their phase 2 push. Really interesting application there, based in Finland. Got a bunch of money there. And then we were able to have a chat with Ken Greenberg. Really interesting application there of non-viral gene delivery that doesn't use an LNP but uses ultrasound, not sonoporation. If you've heard of sonoporation and thought that sounds kind of invasive, this is a much more attractive relative that is much more safe. So there's going to be some really nice little sound bites we'll be able to pull out of that for sure. And then Wise Therapeutics, super cool to see some applications of astrocytes inside of MS applications, and then they've got a nice platform for other major diseases as well. So
yes, sir.
It's been a good week inside of the early stage biotech. Right on.
Sounds good.
Excellent. All right, folks, we're going to leave it there. Thank you for coming, and we will be live once again on Thursday. We're going to have a company coming called Base Immune, and they are developing vaccines for fibrosis, which I never really expected to hear. So if you want to tune back in on Thursday, we'd love to have you. All right. That's it for now. Thank you so much, everyone, and we'll see you next time. Cheers.
Bye.
Bye.